Gentlemen and ladies, welcome back to this series of lectures on excitable tissue. We've been looking at membrane potentials to create a base on how we are going to discover the physiological functions and characteristics of nerve tissue. Um, we begin looking at the objectives. Basically, at this level, all we want you to do as a student is to be able to classify neurons by their function and the anatomy in terms of the histology. Then we also expect you, after this, you should be able to list and explain the role of support cells in nerve tissue, classify synapses, and be able to tell the difference between amongst the neurons in terms of their features as well as their physiological characteristics. Excitable tissue, we have discovered, is tissue that is able to generate and propagate action potentials. So neurons fall into that category of types of tissue. And we have discovered how, because of this characteristic, neurons are able to control other tissues because they use action potentials called electrical impulses to be able to affect the cells electrically. The support cells that are found in nerve tissue, they support in different ways. Because they provide support, literally they are called glue cells, like they are the ones that glue the functionality of nerve tissue together. So oligodendrocytes, as the first example, together with Schwann cells, they produce myelination for nerve tissue. So they provide myelin for axons of neurons. Oligodendrocytes provide myelin or myelination in the central nervous system, while Schwann cells provide myelination for axons in the peripheral nervous system. The other difference is that oligodendrocytes, each one of them will be associated with at least 2 neurons or axons, while every Schwann cell is only associated with 1 axon per time. Astrocytes are very, very important. Astrocytes are very, very important in providing control in terms of nerve environment. So they control materials that flow into nerve tissue and control how nerve tissue allow exchange of materials with the rest of the body systems. This is as a result of their anatomical shape as well as the anatomical features of their feet processes extending to wrap around capillaries of the circulatory system, such that they provide extra layer on circulatory environment of the nervous system. Therefore, they control and limit the movement of material into nerve tissue by contributing to what is called the blood-brain barrier. So this is what we are seeing here. We see oligodendrocytes, we see Schwann cells, we see astrocytes. There are other cells here which are not illustrated, like stellate cells, which tend to be the main source of regenerative capacity of nerve tissue. But we also have ependymal cells, which are important in producing cerebrospinal fluid. We also have not illustrated the microglia cells, which are small cells that are found in nerve tissue, which are activated by stimuli as well as inflammatory environments to be able to become bigger and phagocytose foreign material and help to provide defense or immunity to nerve tissue. Neurons being nerve cells, they come in different types and shapes, but most of them have the same parts that are typical to spinal motor neurons. So we can use a spinal motor neuron as an example of a neuron. to illustrate the features of a neuron. So the neuron will have the soma, or the cell body, and the processes or the extensions. The longer one is the axon, and the shorter ones are the dendrites. The dendrites together with the soma provide receptive capacity of the neuron, while the first part of the axon where it connects to the soma is called the initial segment, where we find the axon hillock, the point where we have regeneration or generation of action potentials or impulses for the neuron. The myelination of the axons, most axons are myelinated to provide efficiency in their function, but also the myelination is not a complete one. It provides nodes, portions where there is no myelination, and these nodes were described by Ranvier, so they are called nodes of Ranvier. And these nodes are the points where we have large populations of voltage-gated sodium channels that allow the large flow of currents of electrolytes. And so this provides the means by which citatory conduction is produced by nerves. So we talked about that in terms of structure. There are processes, there are all these structures that perform different functions on the or in the neuron. In terms of the anatomical features that help to characterize the different types of neurons, we could actually use one simple way to classify neurons based on the number of processes that emanate from the cell body. So neurons anatomically can be classified as unipolar because there is only one process extending from the cell body. They can be classified as bipolar, bi meaning two extensions from the cell body, or multipolar because there are several extensions from the cell body. We've also noticed that some neurons will have one axon projecting, or one process projecting from the cell body, and this divides into two. So this one is called the pseudo-unipolar. It's a fake unipolar. In some literature, it is classified amongst the bipolar, but basically it's a pseudo-unipolar. It's a unipolar, but it is fake because its process tends to divide into two. So some examples of multipolar neurons, we have the motor neuron of the spinal cord found in the anterior horn of the spinal cord. And then we also have pyramidal neurons in the hippocampus, as well as the cerebral cortex, the motor areas of the cerebral cortex. We also have Purkinjes found in the cerebellum. You can see there's the extension of the so many processes, and this improves efficiency in terms of receptive capacity of the neuron. Some of the types of neurons in mammalian nerve cells, we've talked about unipolar. These are found in invertebrates, but the pseudo-unipolar is our main sensory neuron in the peripheral nervous system. Bipolar neurons are found in the retina of the eyes. Pseudo-unipolar, we've talked about sensory system. Multipolar, they are found in so many locations. The functional classification of the zones of the neuron. A typical neuron will have the receptive zone, where we have the dendrites and the soma. Then we have the axon hillock, where action potentials are generated or electrical impulses. Then we have the axonal process, the longer process, the longer expansion, where we have transmission of impulses from the cell body to the terminal. But also the axon is important for transportation of materials to the terminal and away from the terminal. The nerve endings are the point of contact for the neuron to another neuron or to another tissue cell. The cell body is located mostly in the dendritic zone, and this cell body really is the center of where everything takes place, meaning all the functional processes take place. So metabolism, growth, and all these irritability, all the functional physiological processes. Myelination is crucial, and this is produced by oligodendrocytes and Schwann cells. In terms of the chemistry, myelin is a protein lipid, okay, protein lipid complex that is wrapped around the axon in terms of sheets. So it will be wrapped over 100 times, covering that axon to improve its efficiency. So what we find is that there are points where there will be no sheath, and these will be called the nodes of Ranvier, and they are periods of about 1 micrometer constrictions around 1 millimeter apart, and these are important in terms of ensuring saltatory conduction of impulses. And so what we find is that when neurons lose their myelin, they lose efficiency, and we find these in autoimmune conditions like multiple sclerosis, where myelination is lost and the neurons begin to discharge uncontrollably, and we begin to have poor conduction of impulses. Axonal transport, this is transportation of material in the axon. We've seen that the axon physiological functions is not only to transmit impulses, but also to be able to allow materials to move within the cytoplasm from the manufacturing center, which is the soma, to the terminal, and also from the terminal for recycled material, for materials needing recycling back to the soma. So in terms of transportation, there is a retrograde and anterograde transportation. So anterograde transportation, that is forward axonal transport, which is about 400 millimeters per day, and slow transportation back to the terminal, which is slower, about 200 millimeters per day. So let's classify synapses. So neurons tend to connect one to the next to ensure that there is neurotransmission or transmission of impulses from one neuron to the next. And these synapses are classified depending on the anatomy, how they connect neurons, and also depending on how impulses are transmitted, that is physiological. So the classification of nerve synapses is that we base on how one neuron connects to the next one, and what we see is that one neuron can connect to the dendrites of the next neuron, can connect to the dendritic spines of the next neuron, can connect to the soma of the next neuron, even to the axon. Therefore, we will have anatomically axospinous, axonic, axodendritic, axosomatic synapses. So that is very, very important. Then in terms of the physiology, in terms of how impulses are transmitted, the synapses can be classified in terms of chemical synapses or electrical synapses, where the chemical synapses, there is a anatomical junction where there is space between the two neurons, and a chemical has to be released that stimulates the next neuron or inhibits the next neuron to ensure transmission. In electrical synapses, what we tend to notice is that the transmission of impulses in electrical synapses is ensured through the direct sharing of cytoplasmic electrolytes, where one neuron will allow the diffusion of positive ions into the next neuron, and because of this, then the next neuron becomes highly positive, and that is basically depolarization, that is excitation. Therefore, what we will tend to see is that we will have electrical impulses transmitted from one neuron to the next through what are called gap junctions. And the gap junctions will be the means by which the cytoplasm will allow electrolytes to be transmitted. And so we are seeing that there will be two types of synapses physiologically, one needing neurotransmitters or chemicals, and one needing direct electrical charge sharing. So one is chemical synapse, the other one is electrical synapse. So basically, this is where we should end, and these are just the illustrations that you need so that you can appreciate what we are trying to discuss. Otherwise, we meet you in the next lecture. Thank you very much.